In this lecture, I'm going to talk about mast cell, and as you may know that mast cell is a granular type of cell, and it is a tissue cell. Mast cell originate from myeloid progenitor from the pluripotent stem cell. And as can be seen here, these membrane bound receptors, they are high affinity Fc epsilon R1. They are specific for the crystalloid fraction of IgE antibodies. So these are IgE antibodies. So they bind to Fc epsilon R1. And the mast cell, when get activated, actually these are vesicles containing mediators, and these are primary mediators. So following activation of the mast cell, these vesicles, they degranulate, and mediators, they come out. These mediators are histamine, platelet activating factor, eosinophil chemoattractant factor, neutrophil chemoattractant factor, and serotonin, and also heparin may come from the mast cell. And these mediators, actually, they are very important for inflammation. And as you know that inflammation is an innate immune response. So during inflammation, three major events take place, and these are increased capillary permeability, vasodilation, and influx of cells. Actually, histamine is responsible for increased capillary permeability in the blood vessel and the vasodilatation. So histamine here, it is very important for the inflammation, for the improvement of the circulation during inflammation. And also, and this actually, they have, as can be seen here, mast cells, they have receptors for C5a, C3a, and these are anaphylotoxins. These are produced during classical activation of the classical pathway of the complement, and they are responsible for influx of cells and release of the histamine. Other mediators, they come from the wall, arachidonic acid. These two mediators, leukotriens and prostaglandin, and these actually lead to bronchospasm during asthma. So mast cell express high affinity receptors specific for IgE, and they express mediators, primary and secondary mediators, and also they express proteoses. These proteoses may recruit macrophage, may recruit monocytes, neutrophils, and maybe eosinophils to the area in case of parasitic infection. As can be seen here, these are cells that can be recruited due to this or production of proteases. And actually the mast cell is a tissue cell, but the counterpart of the mast cell is a basophils, which is in the circulation. But however, they produce more or less similar mediators. For example, platelet activating factor, histamine, eosinophil chemoattractant factor, neutrophil chemoattractant factor. But however, they differ in their location. And also mast cell interact with T cell, either T helper cell or T cytotoxic cell. And as a result of this interaction, the mast cell will produce interleukin number 6, which it has immunoregulatory effect and promote T cell survival and limit the apoptosis. And this interaction between mast cell and the T cell, it takes place through the major histocompatibility and TCR, and in case of T cell B7 and CD28. Similarly, mast cell has the capacity to interact with B cell through these molecules, the major histocompatibility class 2 and the B cell receptor and CD40 ligand on the surface of the mast cell and CD40 on the surface of B cell, and this also will lead to liberation or expression of interleukin number 6 and interleukin number 4 from the mast cell, and this actually it has a modulatory effect on B cell and promote proliferation of B cell and survival of the B cell. As can be seen here, this is a mast cell, so the mast cell, it can kill some pathogens as they express toll-like receptors on their surface. So they express toll-like receptor number 2, 3, 4, 5, 7, and 9. And these receptors are known pattern recognition receptors. They recognize pathogen associated molecular pattern like peptidoglycan, LBS, and other associated molecular patterns. So as I mentioned, that they produce proteases. These on their own can kill, or they can recruit other cells to kill the microorganisms. Similarly to way of killing by eosinophil, macrophage, neutrophil, basophil, mast cells they have the capacity to modify the chromatin, and the chromatin will send or produce fibers or filaments. These are very sticky, and it has the capacity to deal with microbes, viral infection, and other infection. But however, here, as can be seen here, these extracellular traps, so the mast cells, they have the capacity to produce extracellular traps. Similarly, other cells similar to the mast cell, for example, macrophage, they have the capacity to produce extracellular traps. Eosinophils, they produce extracellular traps. Basophil or bets, they produce bets or basophils extracellular traps. And neutrophils, they produce nets or neutrophil extracellular trap, a process known as netosis. And actually these fibers or sticky filaments, they have the capacity to capture the microbe, and following that the cells will phagocytose these microbes together with a myeloperoxidase present here and reactive oxygen species that has the capacity to kill the microbes. Thank you very much, Professor Ahmed.